Welcome to the Mcat Basics podcast, brought to you by the physicians at Med School Coach. Each week, Sam Smith breaks down high-yield Mcat topics so you can score as high as possible on test day.
Hello, I am Sam Smith, and this is Sam's Mcat Basics. This podcast covers the most important topics put out on the AAMC Mcat each year, and I determined this list by going through all the official practice materials that the AAMC puts out, and also some third-party practice material, and just put together this big list of topics that consistently kept on showing up, and ranked those topics and made podcasts out of it. So this podcast covers a few other topics within metabolism. If you haven't listened to the previous podcast to this, that podcast went over the three main parts to metabolism that you should know for the MCAT, which are Krebs cycle, the electron transport chain, and glycolysis. This podcast covers more into metabolism, but a bit more subtle topics. I'm going to cover gluconeogenesis, the Pentose Phosphate Pathway, and then talk a little bit about ketone body generation, or ketosis. So again, these topics are a little bit less covered on the MCAT, but still very important, and I think you should know them. So here we go. I'm going to start with the Pentose Phosphate Pathway. The Pentose Phosphate Pathway has two main functions. The first is that it converts glucose 6-phosphate, which is a 6-carbon sugar, into ribose 5-phosphate, which is a 5-carbon sugar. And this 5-carbon sugar is an important component of many biomolecules, including ATP, NAD, CoA, DNA, and RNA. And then the second main function of the pentose phosphate pathway is to produce NADPH. So that sounds like NADH, but it is completely different, NADPH. So what is NADPH? I'm going to talk about that first. NADPH has a bunch of different functions. It's really vital in your body. It acts as a reductive agent in cellular respiration. That means it's oxidized, and therefore it acts as an electron donor. And so it also aids in biosynthesis of different molecules. It aids in cellular bleach production and also maintains your natural antioxidants. So to summarize NADPH's function in one sentence is that it aids in biosynthesis reactions by transferring electrons. And if you're like me at this point, you're going to be asking, what is the difference between NADPH and NADH? Well, obviously, a phosphate group is the main difference, which is that P, but less obviously, NADPH is used more in anabolic reactions, so that's like building nucleotides, and NADPH is used in more catabolic reactions or breakdown of glucose, for example. And it's kind of interesting to know that NADPH functions in a very similar manner to NADH. Remember, NADH in our bodies is used as this high-energy electron carrier for metabolic processes. And in photosynthesis, plants use NADPH very similarly to how we use NADH. Kind of interesting. Not much on the MCAT about photosynthesis, but still an interesting thing to know. All right, so getting back to the pentose phosphate pathway. I want to touch on the parts of the pentose phosphate pathway that are the most important and that I think you should know for the MCAT. So what I would not do for this process is memorize all the steps, know the enzymes, know the intermediate names, etc., etc. What I would know if I were you is know that essentially what's going on is that the pentose phosphate pathway is taking glucose 6-phosphate. And if you remember, that is the second step of glycolysis, where you add the phosphate group and activate glucose. So it's taking that glucose 6-phosphate and then processing it, or taking that and then making these two molecules, which is NADPH and also these precursors to these five carbon sugars, the precursors to the 5 carbon sugars. Another important thing to know is the rate-limiting enzyme of the pentose phosphate pathway, and that is glucose 6-phosphate dehydrogenase. Again, the two most important things you need to remember are that the pentose phosphate pathway, number one, it uses glucose 6-phosphate, which is the second step of glycolysis. And number two, you need to know that the rate-limiting enzyme of the reaction is glucose 6-phosphate dehydrogenase. All right, so next, I want to get into glucose homeostasis. And so I'm going to talk a little bit about how your body maintains pretty even levels of glucose in your blood at all times. And then I'm going to get into what happens when your body is very low on glucose. What does it do in order to either make it, or what other forms of energy can your body run off of? And also, I really hope you just checked your phone, even though that was mine that went off. All right, so your body regulates your blood glucose between about 70 to 130 milligrams per deciliter, which is a pretty tight window, but your body keeps it in between these levels. And it does this using two different hormones. And the first hormone, and you've probably heard of these, is insulin. The second is glucagon. And so first I'm going to talk about which cells produce these, and then I'm going to get into their effects. So let's just say you ate a big meal, a Big Mac and fries and a shake, and that's subjecting your body to a pretty big influx of glucose. And because of that, your blood glucose levels start to rise. In response, your body releases insulin, which is produced by beta cells in the pancreas. And this insulin causes the insertion of GLUT4 transporters into cell membranes. And so these are just glucose transporters that enable the glucose to enter cells. And I'll get more into the effects of insulin later. Just know that number one, it's produced in beta cells in the pancreas. And number two, causes the insertion of GLUT4 transporters into the cell membrane. When glucose is gone or blood glucose levels fall, your body releases glucagon. And you can remember this by thinking glucose is gone, glucagon. And glucagon is produced by alpha cells in the pancreas. And it basically stimulates your body to produce glucose in some form. And I'm going to get into what those are exactly a little bit later in the podcast. All right, so let's talk about the biological effects of insulin and glucagon. I'm going to start with insulin. So insulin increases glucose uptake. It increases glycogen synthesis, which is the stored form of glucose. It increases protein synthesis. It increases fat synthesis. It decreases gluconeogenesis, which I will get into. It decreases glycogen mobilization, and that's just the breakdown of your stored glucose pools. And then it decreases lipid mobilization, breakdown of fats, and it also decreases protein degradation. So it's also important to note that insulin exerts its control partially through gene expression. All right, and glucagon does pretty much the exact opposite. So it decreases glycogen synthesis, decreases protein synthesis, decreases fat synthesis, decreases all the synthesises. Or is it pronounced synthi? I don't know. Anyways, insulin also increases gluconeogenesis, glycogen mobilization, ketogenesis, which is another thing I'm going to talk about in a little bit, and increases protein degradation, and also increases the uptake of amino acids. And so you might be asking, why exactly does glucagon increase the uptake of amino acids? Well, as I'll get to in a little bit, amino acids can be used either in the production of ketone bodies or in the production of glucose via gluconeogenesis. And I'm going to get into that, but first I'm going to talk a little bit about glycogen. How is glycogen formed, and then how is a glycogen broken down? So glycogen is made through a process known as glycogen synthesis, and it is an endergonic reaction which means it requires the input of energy. So this process then obviously must be paired with something that is highly exergonic, or something that produces a lot of energy in order to actually occur within your cells. And so the reaction that is paired with glycogen synthesis is the hydrolysis of UTP to UDP. And so UTP is very similar to ATP. They're both high energy nucleoside triphosphates. And I have a podcast that's on biomolecules and biomolecule structure. It might be out by the time you're listening to this. If not, just know it's coming out and you can learn more about nucleoside triphosphates. So you are pairing this removal of the phosphate group from UTP to UDP, pairing that with your endergonic reaction to then push forward the synthesis of glycogen, which again is the stored form of glucose in humans. All right, so here's how this works. So you start with glucose, and the enzyme hexokinase, if you remember this from glycolysis, adds a phosphate onto the 6th carbon, so it forms glucose 6-phosphate. And then this glucose 6-phosphate is converted to glucose 1-phosphate by phosphoglucomutase. And then this glucose 1-phosphate is converted to UDP glucose via a reaction involving UTP. So the alpha phosphate of UTP, which is the one closest to the carbohydrate ring, reacts with the phosphate group already attached to glucose. And again, this is the glucose at the 1 position, or the number 1 carbon. And the result is UDP glucose and pyrophosphate, which is basically 2 linked phosphates. So it's PP. Which is, I'm not going to make a joke about that because I'm above that, but it's PP. And this reaction is catalyzed by a very long-named enzyme called glucose 1-phosphate uridyl transferase. Uridyl transferase. Long word, sounds a little funny. But once you have this UDP glucose, it essentially acts as a monomer, and you can keep adding these UDP glucose monomers onto each other and create these chains of glucose, which are then stored as glycogen. And again, this polymerization is driven by the cleavage of UDP. And so it's a little more complex than this, obviously. There's a few different enzymes you should know. The first is called Glycogen Synthase, and this forms linear chains of glycogen, which are alpha 1,4 linked chains. And you also have an enzyme called the glycogen branching enzyme, and this forms branching chains. So these form alpha 1,6 carbohydrate bonds. So these are what create those branches off this main linear chain. All right, so now that you have a little bit of the basics on glycogen synthesis, and keep in mind that I'm really just kind of hitting the main basis here in terms of what you should know. I'm not getting super into depth, but the MCAT's not going to get super into depth with you. So just, you might get, and this is how I was very, I was thinking, okay, I want to know every single little detail I can. But in reality, you really want to just study. You want to know kind of the main points of each topic, because the chances of them asking you these really random questions are, number one, very slim, and number two, the odds of you actually studying this little teeny nitpick detail that you see on the MCAT are probably pretty slim too. All right, so on to glycogen breakdown. So glycogen is cleaved from the non-reducing ends of the chain by the enzyme glycogen phosphorylase. So non-reducing refers to the ends of the chain that contain the carbon that is only attached to one oxygen. So if you remember from organic chemistry or even biochemistry, the reducing end of the reducing end of the glucose molecule is the one that contains that carbon that is attached to 2 oxygens. And it's called reducing because that's what somebody named it. No, just kidding. I don't know why it's called reducing end, and I don't know. I don't think you gotta know that. And if you have no idea what I'm talking about in terms of the non-reducing end and the reducing end, go look that up. It's pretty simple. So again, Glycogen is cleaved from the non-reducing ends of the chain by the enzyme glycogen phosphorylase, and this cleaves glucose molecules off the linear chains. These, again, are alpha 1,4 bonds, and this produces monomers of glucose 1-phosphate. So then what happens to these glucose 1-phosphates? Well, these glucose 1-phosphate is then converted to glucose 6-phosphate by phosphoglucomutase. This is just the opposite pathway of what I described before. And then this glucose 6-phosphate can then either be directly fed into the glycolysis pathway, it can be turned into glucose via dephosphorylation, or it can enter the phosphate, the pentose phosphate pathway. So it's got a lot of different options. You just got to pick whatever you want. Pick your fancy. And so I know you definitely are probably thinking, well, what about those alpha 1,6 branched chains that you were talking about earlier? What happens to those? Well, a special enzyme is required in order to break those, and it is called a debranching enzyme. And a glycogen debranching enzyme works like this. So it breaks down. You're gonna have first of all, you have these long branches off the linear glycogen chain, and your debranching enzyme is going to break off these monomers, these glucose 1-phosphate monomers, in the same way that the linear enzyme is going to break it down. And then once you get to the last 4 glucose molecules in the branch, this enzyme is going to take those and it's going to transfer those. It's going to transfer 3 of those to the linear chain, and then it's going to break off that last one. And so essentially what happened is you just moved 3 of the last 4 residues of the glucose residues onto the linear chain, broke off that last one, and now your glycogen phosphorylase can break down those glycogen or those glucose monomers off. So again, that was probably a little bit confusing. The last 3 of them, the last 4, you take 3 of those, add them onto the linear chain. That very last one, it breaks off with a debranching enzyme, and then the 3 that you transferred are then broken down into monomers by the glycogen phosphorylase.
In general, the thing I always try to say to our students, and certainly prospective students, people desire the product coming out of the DO schools. And we hear very, very positive feedback from program directors and institutions that for a long time have been accepting DO students, and certainly the stories about them becoming leaders and practitioners in those institutions.
That was a clip from the Prospective Doctor podcast with the CEO and president of the American Association of Colleges of Osteopathic Medicine, or AACOM. The Prospective Doctor podcast is great. It features weekly interviews with interesting doctors and different figures in medicine. If you're a pre-med or medical student, I highly recommend it, and I really do listen every week, and I feel like I'm learning something new and valuable with each episode. So if you're looking for a new podcast, go search Prospective Doctor podcast in your podcasting app and take a listen. All right, so the last 2 things I want to talk about here are gluconeogenesis and ketone body generation, or ketosis. So gluconeogenesis is a process in which you create glucose from other non-carbohydrate carbon sources. And so these carbon sources are fatty acids, amino acids, lactate, and so gluconeogenesis, in terms of amino acids, you can only use the glucogenic amino acids for gluconeogenesis. And the glucogenic amino acids are everything other than leucine and lysine. So remember that, have that memorized for the MCAT. The glucogenic amino acids, which feed into gluconeogenesis, are everything other than leucine and lysine. And this process uses the reversible enzymes from the glycolysis pathway. And still, though, it must get past the 3 irreversible steps of glycolysis. These are like roadblocks. You can't use these enzymes to go forward, you can't use those enzymes to go backwards in the process. So you must bypass those. And so this is how you bypass them. So starting here from the end of glycolysis, the first roadblock you need to get through is the pyruvate kinase. And so gluconeogenesis uses pyruvate carboxylase, which converts pyruvate to oxaloacetate to get past that irreversible step. All right, so the next enzyme that you must get past in glycolysis is phosphofructokinase. And so to do this, you use an enzyme called fructose 6-bisphosphatase, and that takes fructose 1,6-bisphosphate and it converts it to fructose 6-phosphate. So if you remember from glycolysis, phosphofructokinase went the opposite way. It converted fructose 6-phosphate into fructose 1,6-bisphosphate. And lastly here, the last hurdle we need to jump over in this process is the enzyme hexokinase. And so hexokinase, if you remember, adds a phosphate group to the glucose and kind of activates it. And so you use, in this case, the enzyme used to bypass this process is an enzyme called glucose 6-phosphatase. And that converts glucose 6-phosphate back to glucose. And boom, you're at glucose. And that is how your body essentially takes this pyruvate, or takes all these carbon sources and converts them into glucose. And obviously I skipped a lot of steps, but just remember you can use a lot of these different non-carbohydrate carbon sources, feed them back into a lot of these glycolysis enzymes, and a few that aren't glycolysis enzymes, and you can get back to making glucose. And like a lot of the other processes I've talked about, there's quite a bit of regulation. And so in terms of the molecules that upregulate gluconeogenesis, you have acetyl-CoA and citrate. And so those two molecules upregulate gluconeogenesis. And so you may be thinking or asking, why do these molecules upregulate gluconeogenesis? Well, essentially, they both act on enzymes in the gluconeogenesis pathway. So acetyl-CoA activates pyruvate carboxylase, and then citrate activates fructose 1,6-bisphosphatase. So on the flip side, gluconeogenesis is downregulated by low energy molecules, and so it's downregulated by AMP and ADP. AMP, ADP. And the enzymes that are regulated are these enzymes that are used to bypass the irreversible steps of glycolysis. All right, well, the last thing I want to touch on is something that you have probably heard in terms of a diet. I'm sure you know a Dave or, a Brad who's really into CrossFit. He's got long hair, most likely wears it in a man bun. And, Brad just won't quit telling everyone how much he loves the keto diet.
Oh, yeah, brother. I've been on the keto diet since 1991.
So, what is ketosis? Well, your body starts to generate these ketone bodies after periods of prolonged starvation, and this is usually after gluconeogenesis occurs. And so after your body has been using the gluconeogenesis pathway, it diverts a bunch of oxaloacetate from the liver, and after a while this leads to a buildup of acetyl-CoA, and then this acetyl-CoA is then used to generate ketone bodies, which is why this usually occurs after gluconeogenesis. And these ketone bodies come in two forms. So number one is called acetoacetate, and the second is called beta hydroxybutyrate. And these are generated through kind of a complex reaction. Just know that ketone bodies partially come from ketogenic amino acids. And this, if you remember, is lysine, and these are lysine and leucine. Ketogenic amino acids, lysine and leucine. So these amino acids can be directly degraded into acetyl-CoA, and then acetyl-CoA is used down a pathway, boom, 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 down this pathway to eventually create a ketone body. And these ketone bodies circulate around your body, and cells in your brain and your heart, etc., use these ketone bodies for energy. And of course, again, this is after very long periods of starvation, when you've depleted your glycogen stores and you have gone through a lot of gluconeogenesis, which is why most likely your friend who claims that they were on the ketogenic diet, probably not the case. And all right, this concludes podcast 4B, which was on metabolism, kind of a mishmash of topics, kind of random, but nonetheless important. Hope you got something out of it. Look forward to our next podcast, which is going to be the 5th in the series, and that is going to be on biomolecule structure. It's going to be a big one, very important. Take a listen. Thanks as always. All right, so now for the segment called Mcat Advice of the Day. So what I'm recommending today is that part of your studying process, what you should do is download a bunch of random journal articles, print them all out, keep them in a folder or something, and, whether it's once a week or even once a day, go through. You don't have to read the whole thing. Read sections of it and try to be able to understand what they're talking about. So the way the MCAT is kind of formatted is it's like reading a random scientific paper. So the more you're able to read them, stay focused on what you're reading, and be able to analyze them, the better. So when I was studying, I would take Wednesday mornings and I'd do these readings where I just read a bunch of papers, and it gets really boring, but the whole point is to kind of train yourself to, even when you're bored, keep reading and be able to pull out information. So that was good practice for me. Something you can incorporate into your studying as well.
Each episode of MCAT Basics is brought to you by Med School Coach. To access Med School Coach services, including MCAT tutoring and medical school admissions advising, visit our website at medschoolcoach.com. Good luck as you prepare for the MCAT, and we hope you tune in again next time.
Come on, Murph. I'm trying to podcast over here, dude.